A very good evening, everyone. I'm Sandhya Shekhar. So, I will be doing the TA duty for your PT 39 bioreactors course, and this is week one course. Hope you guys have gone through the lectures that was given by Professor J.K. Suresh Kumar. And today we will be solving the tutorial problems of week one. This will really help you in solving the assignments that are posted for week one. So we'll get into the tutorial at first. So let me share my screen. Yeah. So we will be seeing tutorial 1 of BT39 course, that is bioreactors. It's a 4-week course and we are in tutorial 1, that is related to week 1 course. So we'll get into the first question. Which options best describes hybridomas? Option A, antibody producing cells in the body. Option B, cancerous cells in the body. Option C, a fusion of antibody producing cells and cancerous cells. Option D, cells producing low cost insulin. So we'll just see like what the hybridomas are, and then we will come to each and every options, and then we will rule out which are not the right answers, and we will choose the one that is right at the end. So first we'll see what is hybridoma. What is hybridoma technology? So in hybridoma technology, we have to produce antibody producing cell, and that is a cell which can produce antibody at a very high concentration, or like it should comparatively what is happening in our body is like if any foreign particle enters our body, our immune response will get elicited and our T cells will be producing our B cells will be producing our antibody of interest that is particularly against the foreign particle that has entered the cell. So that the the antibody produced by the B cells uh, will target the foreign particle that has entered our cell. But what to say, B cells and whatever these are very short lived cells. Okay, it can't replicate or it can't sustain for a very long time. So hybridoma technology was designed in such a way that these B cells are modified in such a way that it can the lifespan of this cell can be like very long as well, and then it can produce the antibody of interest that can target your foreign particle, like specified foreign particle. So how this technology works is like antigen is injected to elicit immune cells. So we can't do these experiments in human level, right? So whatever the model organisms are there, which are like very closely related to humans, one such is rat. So the antigen, that means whatever the foreign particle I told, right? The foreign molecule that we call it as antigen. So when this antigen is this injected in such a way that it has to elicit only the immune response, the concentration will be very fixed in this. We can't give very high concentration, that might even lead to the death of the mice also. So how it has to be done is like it has to be given in very less concentration such that it just elicits the immune response. Okay. And the plasma cells are isolated from the spleen, from there it is being isolated. So plasma cells are nothing but if you see the immune response pathway, B cells are nothing but these are being differentiated by the plasma cells. So the plasma cells will get differentiated into B cells, and B cells will give rise to antibodies. So that's how the pathway comes. So the plasma cells are being isolated from the mice model, and this, as I already told, plasma cells are short-lived. So we have to make sure that this plasma cells are going to have a very long lifespan. For that, what has to be done? So what we know about cancer cells are like these are very fast. The what to say, the replication rate is very fast for these cells. It can replicate very fast, and these cells are very long lived. That's why cancerous cells are being selected for this purpose. So the myeloma cells are being isolated, or it can be taken from pure culture. So these plasma cells and myeloma are like combined to form the hybridomas. So these are fused. The plasma cells and the myeloma cells are fused to produce hybridomas. So like this will be further like selected on a medium. So we will not know, right? Like once the fusion is being done, it can be either plasma cells, or it can be myeloma cells, or it can be a fusion of plasma and myeloma cells, or fourth option can be a plasma cell and a plasma cell can be fused, a myeloma cell and a myeloma cell can be fused. So totally we have 5 possibilities. So what will be done here is like it will be selected on a hat medium, so hypoxanthine, adenine and thymidine medium, in which these plasma cells, as we already know that these are short-lived cells. So even if a plasma cell and a plasma cell is being fused, that is not going to survive for a long time, because the experiment will be incubated in such a way that whatever the cells are long-lived, that can be only picked for further selection. So here plasma cells, even if it is being there on the medium, these are short-lived and it will not sustain after a long time. Whereas myeloma and myeloma cells will be fused if those are available on the hat medium. It will be eliminated. If you read about hat medium, you will get to know a lot about that. This medium is selective in such a way that only plasma cell and myeloma cells which are being fused can only be that can only sustain on this medium. Okay, so we'll come to the options. Option A, antibody producing cells in the body. No, as we already told, the B cells that are those are the antibody producing cells in the body, right? These are short lived and these are not categorized under hybridomas. Whereas cancerous cells in the body, just a minute, I'll just take my marker. Yeah, cancerous cells in the body. So these are not going to give rise to hybridomas. Option C is a fusion of antibody producing cells and cancerous cells. So fusion of, that's what we are seeing here, right? Antibody producing cells are B cells, but as I already mentioned, those are B cells are being differentiated from the plasma cells only. So plasma cells and the cancerous cells, so nothing but myeloma. So a fusion of these two only will give rise to hybridomas. So option C is right answer. Whereas cells producing low cost insulin, no, these are not related to your hybridoma cells at all. So the right answer is option C, a fusion of antibody producing cells and cancerous cells. Moving to question 2, which of the following products are commercially produced using bioreactors? So option A, insulin, milk, petrol, bio-oil. Option B, insulin, MABs, antibiotics, biomass. Option C, bread, milk, bio-oil, curd. Option D, MABs, insulin, crude diesel, bio-oil. So the question is, which of the following products are commercially produced using bioreactors? Okay, so bioreactors are nothing but, we can do most of the experiments in the shake flask level also, but in bioreactor, why we have to go for bioreactor? If such a question is being asked to you, the first answer that has to come out of you is that to maintain a controlled environment, like you can't maintain the oxygen level as well as pH and all these important bioprocess parameters in shake flask level. Okay, at all the points, like throughout the fermentation, you really can't maintain these conditions in the shake flask level. So that's why people come for bioreactors, where a controlled environment will be given for the cells to produce a product of interest at a very high concentration, rather than producing the cells at like 2 liter or 3 liter flask level. Okay, so we will just see the options and we will try to rule out the options which are not correct. Okay, yeah. Insulin. So we all have already studied that insulin, there are many recombinant cells that are being used in the bioprocess industries to produce insulin as one of the major products, pharmaceutical products. So we can say yes, insulin is being commercially produced using bioreactors. Milk. Whether milk, I'm not very sure. Sorry, I'm very sure that milk is not being produced by the bioreactors, but there are many dairy industries which are producing milk proteins like casein and all these renin and all these milk dairy industries, they are producing milk proteins using commercially using bioreactors. So that is true. But if you just say milk, so that is a wrong answer. Like milk is not being produced, commercially produced using bioreactors. Petrol. If it's anything related to biodiesel or else like if the word bio has been present, like FAME, we call something as FAME, fatty acid methyl esters. So the biodiesel as well as whatever the bio oil that is being produced, that are produced using the uh, FAME products only, like triacyl glycerol to what to say, your fatty acid methyl esters are only derived. But if you see petrol are just crude components which are got from mining. Okay, these are not being commercially produced using bioreactors. Yes, the other option is oil. Bio oil is being produced, as the word bio itself is already there. Yes, bio oil is being commercially produced using bioreactors. As I have already mentioned, there is something called triacyl glycerol. So in this, three glycerol components will get attached to fatty acid of different chain lengths. If we treat it with methanol, this will give rise to fatty acid methyl esters. So these are categorized under bio oil. So option A is not the right answer because out of four options, like two are wrong. Coming back to option B, insulin, as we already mentioned, yes, insulin is being commercially produced using bioreactors. MABs, these are called MABs. These are nothing but monoclonal antibodies. So monoclonal antibodies are like kind of biosimilars, or else like you can categorize it under biosimilars, or else like monoclonal antibodies are being produced using both mammalian cells as well as like microbial cells in at a bioreactor level in many of the industries, because these are therapeutic products which are having very high commercial importance, and these are mainly for therapeutic purpose. Like each and every monoclonal antibody will be specifically targeting to cure a particular disorder or a disease. So yes, MABs are being commercially produced using bioreactors. Antibiotics. Yes, we can say that antibiotics are commercially produced using bioreactors, because we already know that antibiotics are during World War and all, we used to read, right? Penicillium notatum has produced penicillin, the antibiotic. So many antibiotics streptomyces and all these genus are commercially used in the industrial scale to produce antibiotics of a of specific antibiotics. So I can say that yes, antibiotics are being, so antibiotics can be either bactericidal or bacteriostatic. So it can, for example, kanamycin, chloramphenicol, and many of these are like antibiotics only. This can either go and prevent further replication, or else like it will just put the replication at a halt. That is bacteriostatic, whereas bactericidal is in such a way that it will completely kill the cell. Cidal is cell, kill. So the antibiotics can be of two types, either it can be bactericidal or bacteriostatic. Coming to biomass, so can biomass be commercially produced using bioreactors? If you have seen the lectures of the professor, you will know that yes, biomass is also a product itself. So in some cases like spirulina and all these things, the biomass itself is a product. So these are being commercially produced using bioreactors, and these are being sold also. Like so, if you see the option B, all four are correct. So option B is the answer. Still, we will just go through option C as well as option D. So option C, bread. So no, bread is not being commercially produced using bioreactors, but the yeast that is being used for like the bread production or something, that can be produced using bioreactors, because biomass itself is a product, right? Milk, as we already told, no, milk is not commercially produced using bioreactors. Yes, oil, bio oil is being produced using the bioreactor, as we talked about triacyl glycerol. Whereas curd is not being produced using bioreactor, but the curdling proteins can be produced using the bioreactor. So you have to really see what has been asked in the question. Be very particular. Don't include that curdling proteins will also come under curd. No, not like that. If that is the case, they'll really mention like what are all the proteins and all. Coming to option D, MABs. So MABs are being produced by the bioreactors, that is monoclonal antibodies. Insulin is being produced by the bioreactors, as we already told. Crude diesel, no. If it's biodiesel, yes, I will take this option. But if it's crude diesel, no. Crude diesel is nothing but whatever we are mining and then taking it from the earth, that is only crude diesel. Those are not being commercially produced using bioreactors. And bio oil is. So make sure, even if you find one option as the right one, make sure the other options are not completely right. We might be wrong at some point. Even I would have, if this has been given as instead of crude diesel, if option D is being given as biodiesel, then this option D will also come very right, completely right now. So you should be very careful while choosing an option. Even if A or B comes completely true, make sure you are checking all the other options, so that you rule out all the other options and then choose only the one which is completely right. Okay. So moving to question 3. The media used for bioprocess is contaminated with single cells with similar thermal response characteristics. It has been observed that by maintaining the media at 60 degrees Celsius for 25 minutes, 85% of the cells become non-viable. Determine the decimal reduction time. Choose the answer closest to your calculated answer. Option A, 12.65 minutes. Option B, 20.22 minutes. Option C, 30.37 minutes. Option D, 25 minutes. So what has been asked is like they are maintaining the media at 60 degrees Celsius for 25 minutes. And what they have observed is that at the end of the 25 minutes, 85% of the cells have become non-viable, which means 85% of the cells are dead. So we have to find what is the decimal reduction point. Okay. So, if we have gone through the lecture by the professor, you will know that decimal reduction point is nothing but the time taken for a 10-fold reduction in the viable cell concentration at a given temperature. So, if you have like 100, say for example, if you had 100 cells, and at the end of 20 minutes, if the viable cell concentration has been reduced to 10, okay, at the end of 10 minutes, for example, I'm saying, okay, if 100 has become 10, then 10 is your decimal reduction point, because 100 divided by 10 comes 10, that is the 10-fold reduction. So 10-fold reduction in the viable cell concentration at a given temperature, whatever the time has taken for this 10-fold reduction, that is only called as decimal reduction point. So for whatever is being asked, we will just find out what is the decimal reduction point. So we know the formula for decimal reduction point. D is equal to 2.303 divided by Kd. Kd is nothing but the thermal death rate constant, right? So we don't know the thermal death rate constant as well, and we have to find out the decimal reduction point, that is D. So first we have to find Kd, right? So we will just see what are all the given data given in the problem. So first thing that we have to notice is that these are single cells, because this is like very the formula can be applied only if a similar temperature is being maintained, the same temperature is being maintained, and these are only for single cells. These are not for a multiple cells or something. Okay, so given is it's being contaminated with only single cell, and what they have given at 60 degrees Celsius for 25 minutes, the viable cell concentration goes from 85% of the cells have become non-viable. Okay, then at the end of 25th minute, what does that mean? Only 15% of the cells are remaining viable, right? So at 0th hour, 100% viable cells were being observed. At the end of 25th minute, when we maintained the temperature at 60 degrees Celsius, the viable cell concentration has been reduced to only 15%. Okay. So if we see the fold reduction, 100 divided by 15, that is equivalent to 6.66 fold reduction has only happened. Okay, so your answer is going to be more than 25 minutes only. Okay, so we will see. To find KD, this is the formula. T is equal to 2.303 divided by KD and into log of xv naught divided by xv. So xv naught is nothing but the initial viable cell concentration, whereas xv is the viable concentration at a specific time. So we'll just rearrange the formula. KD is equal to, just a minute, I'll take a laser pointer. Yes. So we will just rearrange. So KD is equal to, so how it has happened? KD is equal to 2.303 divided by T log of xv naught divided by xv. So we have already found out xv naught divided by xv, that is 6.66. So log of 6.66 is being written here. Whereas 2.303 divided by KD. KD is nothing but your, sorry, here T has been rearranged, right? So 25 minutes, always KD should be in seconds. Okay, so 25 into 60, which is nothing but 1500. So if you just solve this, you get 1.26 into 10 power minus 3 second inverse, because here the unit and unit will get cancelled, because both will be in gram per liter or like whatever 100% was there. So this unit will get cancelled. Only T was there in the denominator. That's why KD is always in time inverse. So it should be always in second inverse only. And coming back to D is equal to 2.303 divided by KD. As we already found out, if we just substitute and then divide, we will get 1821.55 second, and again divided by 60, so that we will get it in terms of minutes. So we have almost gotten 30.35 minutes. So 30.35, where it is nearby? Option C. 30.37 is very near. Okay. So answer is option C. So we can really guess it from the beginning only. For example, if you have gotten an answer which was almost like 15 minutes or 20 minutes, you can easily find out, because at the end of 25th minute only, you're having 6.6 fold reduction. Then how you would have had, like in 15 or 20 minutes, how you would have had a 10 fold reduction? Obviously the 10 fold reduction will come after 25 minutes only, right? So with these small tricks, you will be able to understand, like, be very logical when you get an answer. Think logically whether that answer is possible or not. If I got an answer here of almost like 12 or 15 minutes, I will really not choose the option, because we already know at the end of 25th minute only 6.6 fold reduction is only being observed, right? So that is not possible. Moving to question 4, identify the mass balance equation for the microbial cells in a bioreactor under batch operation. So here they have given Ri indicates rate of input of cells, R naught is rate of output of cells, Rg is the rate of generation of cells, Rd is rate of consumption or rate of death rate of the cells, and dA by dt denotes the rate of accumulation of cells. Okay, so option A, Ri minus R naught is equal to 0. Option B, Ri minus R naught minus Rd, which is like rate of input minus rate of output minus rate of consumption is equal to 0. Option C is rate of generation minus rate of consumption is equal to the rate of accumulation of cells. Option D is Ri minus R naught is equal to Rc, which is rate of Rc is nothing but RC and RD are same here, I guess. Like, that's what I am like saying you guys. This is not RC, this is RD. I'm sorry for the confusion. Rate of conception or rate of death. So this is RD only. Yeah, we'll further go. So we'll see first like what is the given data. So we have been asked for only batch operation, right? So in batch operation, what happens? First we will autoclave the reactor. By putting the media in that, we'll prepare the media. We will just introduce the media inside the bioreactor. We will sterilize the bioreactor, and then once the reactor comes to the room temperature or the specified temperature, for example, if it's E. coli, we have to inoculate at an optimal temperature, that is 37 degrees Celsius. So we'll set up the reactor at the specified optimum temperature, and then we will inoculate the cells in that. So after inoculation in the batch process, the batch process will go for a like if it's E. coli, it might go till like 24 hours or like even 30 hours, for example. So that is dependent on the initial substrate concentration what we have given in the medium. So for example, if it's getting over at 30 hours, we will be collecting media we will be collecting the samples regularly to do the biomass analysis, product analysis, as well as the substrate analysis and all. But once the batch operation starts, there will not be any inlet or outlet of the cells in the and other than taking samples, we will not be removing the media or adding the media for any reasons. Okay, so your input, we will see the equation. So how the mass balance equation is being written: overall mass balance, rate of input minus rate of output, plus if there is any regeneration. Obviously regeneration of cells will be there, right? What will be happening in your bioreactor? You have given nutrients in the medium and you have given an inoculum that will utilize the nutrients, and it will keep on replicating. So replication is nothing but the generation of cells, right? So plus generation of cells, and if there is generation after a certain point, the cell death will also be there, right? So minus rate of consumption. So if you just subtract rate of generation minus rate of consumption, like what it will be like, this is the overall equation, right, which will be equivalent to the rate of accumulation of cells. Okay, so as we have already told, in batch process there won't be any input of cells and there won't be any output of cells also. So your input of cells as well as your outlet of cells will be zero in case of batch operation. Okay, so what it becomes, Rg minus Rd is equal to accumulation. So this is the answer for your question 4. So when it's very normal, right? In a batch operation, you are not feeding any cells inside, you are not taking away any cells inside. So whatever is being replicating inside minus whatever is being dead, that will only lead to the accumulation of cells, right? Like nothing else is going to come in from somewhere else. So whatever is there, rate of generation is the overall cells that are being generated, minus whatever was already dead. Okay, so the dead cells also you have like subtracted. So that only will lead to the accumulation of cells. Whatever is remaining in the media as live cells, that is only the rate of accumulation of cells. So option C is the right answer. Moving on to question 5. Match the following important components of bioprocessing. So recombinant E. coli, glucose, amylase. So we have to find what is the substrate, what is the product, and what is the inoculum. So if E. coli, actually E. coli is being used as this is one of the well-studied organism. So this has been used in industry in a lot of process. So if you see recombinant E. coli, if it has only been written as E. coli, so as it's a cell, we have like two options for this. Okay, this itself can be a product because we know that biomass can be a product, right? And this can be inoculum as well. Can E. coli be a substrate for something? Maybe, but not in this case. Like we say, right, if there is any fungus or something that will feed on the dead biomass. Okay, so if there is any fungi in the option, we can think recombinant E. coli as a option for being a substrate, but that is also not being given in the category. So recombinant E. coli can be a product or it can be a inoculum. But if you want E. coli as the E. coli biomass as the product, why you have to do some recombination in that, right? You just need the biomass. For biomass, no need to do the cloning and no need to do the recombinant part here, as far as I know. Okay, so recombinant E. coli cannot be a product, but it can be given as an inoculum so that it will replicate and produce a recombinant product of interest. So for option 1, E. coli inoculum is the answer. And glucose, coming to glucose, can glucose be a product? Huh? Can glucose be a product, or it can be a substrate? Only two ways are possible. Okay, so here you have what are left out: amylase as well as glucose. Can amylase be a substrate? No. Amylase is an enzyme, so that's what they are going to tell here as a story. Okay, so glucose is being used as a substrate by your recombinant E. coli that is being inoculated into the media, and this recombinant E. coli is going to feed on the glucose, which is used as substrate, to produce the product called amylase. So how it's going to be: 1 gives rise to Y, and 2 glucose. Glucose is nothing but this is a substrate Z, and 3 amylase, that is the product. So Y is at X. So where is the right answer? Y, yeah, Y is at X. Is there any Y is at X? No. So the right answer is B, that is Y is at an X. Recombinant E. coli is an inoculum. Glucose is being used as a substrate, and amylase is being used as the product. Okay. Question 6. The main factors to be optimized while designing a thermal sterilization process. Okay, just a minute. So, the main factors to be optimized while designing a thermal sterilization process are initial pH of the medium, time of exposure, DO, temperature. So, a thermal sterilization process. So, during autoclaving media, is it like the initial pH of the medium? Is it going to matter a lot? Yes, actually, if you just talk about in the perspective of medium, like it will matter a lot, because if you, for example, you're going to work on any minimal salt media, okay, it will contain many salts. If you just autoclave the media as such without adjusting the pH, all the salts might get precipitated. Okay, this is in the perspective of medium, but will it affect the thermal sterilization process? No. Even if your salts are going to get precipitated or deposited, it is going to sterilize only. If you keep it for autoclaving, it is going to reach 121 degrees Celsius and maintain it for 15 minutes at 15 psi. Anyway, even if the salts are being deposited also, the sterilization will happen for sure. Okay, so if you think initial pH of the medium, is it one of the important factors to optimize the thermal sterilization process? No, it is not going to. Whereas time of exposure, so as we already say that thermal sterilization process that we do, like 15 minutes time we do, but what if you see the graph, like what if you do it at 121 degrees Celsius for like 40 minutes at 15 psi. If you have kept a medium, like for example urea broth, uh, it was reported that some amount of nutrients are being degraded. So your, for example, you have used tryptone of like 20 gram per liter, that might get reduced to say for example 18 or 17 gram per liter. So when you start your experiment, there will be a degradation in the nutrients that are already present in the medium. If you increase the time of exposure of during thermal sterilization, whereas if you reduce the time of exposure, what happens? For example, it's like going to be 15 minutes, but if you try to reduce the like time of exposure, then it is going to your viable, whatever the cell concentration is there in the media, that will not be completely destroyed. So your thermal sterilization process will not be completed until and unless you are keeping it for 15 minutes 121 degrees Celsius. So time of exposure matters a lot, and it can be considered as one of the important factors while designing a thermal sterilization process. And option C is dissolved oxygen. So if you consider dissolved oxygen as one of the parameters, during fermentation, DO plays a very important role, because if it's an aerobic organism, it really requires dissolved oxygen. If it's a facultative anaerobe, it may or may not need dissolved oxygen. Whereas if it's an anaerobic process, there should not be any dissolved oxygen that should be present in the medium, right? So DO plays a very important role in terms of fermentation. In the case of fermentation, while designing a thermal sterilization process, how DO is going to matter, because we are not going to cultivate the cells here. We are going to completely destroy the cells. For that, this is not going to be one of the important factors. Okay, so till now what we have seen, time of exposure is being considered as one of the important factors for thermal sterilization process. Coming back to the next option, that is option D, temperature. So, uh, so as is the thermal sterilization process, yes, temperature is going to be one of the important factors, right? Because, for example, instead of keeping it at 121 degrees Celsius, if you're going to put it at 100 degrees Celsius or like 110 degrees Celsius, so at that time you have to increase the time of exposure should be more. It shouldn't be only restricted to only 15 minutes, right? So in that way, temperature plays a very important role. So for this typical death rate data for spores of Bacillus stereothermophilus bioprocess engineering, I have taken this from Schuler and Kargi book. Okay, so if you just see, they have given N by N naught, which is nothing but N naught is the viable cell concentration at the start of the uh, so N by N naught they have given. So if you see, they have plotted it on the basis of two. One is up to temperature, considering temperature, and the other is time. So when they have kept it for 121 degrees Celsius, and the time was almost, they have got 10 power minus 3 N by N naught. In between, like it's almost like we can say 1.5 minutes or like 2 minutes. In 2 minutes only, they have reached 10 power minus 3 N by N naught. Whereas when they have taken like 110 degrees Celsius and they have maintained it almost 12.5 minutes has been taken to reach the same 10 power minus 3. Whereas in the case of 108, this was even more, this was almost 22.5. So what I'm saying is like, that's what I already told. If you want to reduce the temperature, the time of exposure will improve. So from this data, it is very well understood that what to say, your temperature and time are the important factors that has to be optimized while designing a thermal sterilization process. Okay, so when a question is being posted to you, just check. Here they have already given very clearly it is going to be main factors. So don't choose only one option. If there is a possibility of another option, choose that as well. Okay, moving on to question 7, identify the type of bioreactor where the aeration and agitation is brought about pumped air only. So option A, stir tank bioreactor. Option B, airlift bioreactor. Option C, packed bed bioreactor. Option D, solid state bioreactor. We will see each and every bioreactor where only pumped air being brought inside. So coming to stir tank bioreactor. So in stir tank bioreactor, we can see that there is a sparger, right? Which is sparger here, whatever the holes are being present, right? Like this is the sparger. Okay, so the sparger is nothing but that is the way the aeration can enter inside the bioreactor. Okay, air is being passed through the sparger and then it will enter in the fermentation medium. Okay, so here pumped air is not only bringing the aeration and agitation. That is like both. Here sparger only bringing the aeration, whereas stir tank bioreactor means we'll have a Russian turbine here, that is only going to that we are going to use it as an agitator, and it is going to break the bubbles that are being entered through the sparger, and whatever the dissolved oxygen that are being released, that will get dissolved in the media and that will be taken up by the cell. So in stir tank bioreactor, the aeration is being brought up by the sparger, whereas the agitation is being brought up by the agitator only. So here pumped air is not only doing both the activities. Moving on to airlift bioreactor. In airlift bioreactor, as we already know that the sparger is there, that will enter inside the hollow tube, and here the sparger aeration rate will be very high in such a way that whatever the cells are there in the hollow tube, due to the airflow only, that will be mixed by passing to the other hollow tube that are being placed beside. Like this is just only one compartment. So here with whatever the force that the aeration is entering the airlift bioreactor, that only is going to help the cells to get mixed also. So here aeration and agitation both are being brought up by pumped air alone. Okay. So moving on to packed bed bioreactor, here also aeration enters from the bottom. Sorry, 1 minute. Aeration enters from the bottom of the packed bed bioreactor, and then it will go through the it will go as an outlet also. But in packed bed bioreactor there will not be any stirring. As far as I know, there are nothing is going to get agitated here, because the cells will be packed as such, and the aeration, the purpose of sending aeration is only to give aeration to the cells, to give oxygen to the cells. That is the sole purpose of sending aeration in terms of packed bed bioreactor. Okay, so here that is not the right answer. Whereas in solid state bioreactor, there is no stirring at all. Your solid state waste will be kept on the top of a tray, and then the aeration will be sent, and the inoculum will be placed on top of this solid state bio waste. And using the aeration for oxygen, the organism will replicate, and then it will be feeding on the solid state waste that are being kept. So out of the four options, in airlift bioreactor only, both aeration and agitation are brought up by pumped air alone. Moving on to question 8. Dr. A is going to do a microbial experiment in a laminar airflow chamber. Which of the following can she use to sterilize the hood? Option A, syringe filtration. Option B, UV lamp. Option C, 70% ethanol. Option D, autoclaving. So if we see syringe filtration, we just use it for a temperature sensitive chemical or a component, for example, antibiotics and all. So syringe filtration, how will it be useful in sterilizing the whole hood? So that is not the answer. And coming to UV lamp, yes. Before starting any experiment, like if you are going to use a new strain, like you have to turn on the UV for at least 15 to 20 minutes, so that whatever the cells are that are already spilled or present in the laminar airflow chamber, that will be completely killed. Okay, option B is one of the right answers. Coming to option C, 70% ethanol. Yes, we know that 70% ethanol is a disinfectant that will be used to wipe the laminar airflow chamber. So every time when you do an experiment, before and after the experiment, you have to wipe the hood with 70% ethanol. Option D, autoclaving. No, we cannot autoclave the laminar airflow chamber and all. So the right options are option B and option D. So here I have given a explanation. So in UV lamp, there is this wavelength UVC, that is 200 to 280 nanometer. It is highly effective in damaging the DNA by forming thymine dimers. Okay, so if TT is there, it will form thymine dimers. So this will halt the replication process. Okay, the DNA polymerase cannot pass through this thymine dimers, so the replication process will be halted for a long time. When this prolonged cell arrest happens, this will automatically lead to cell death only. Apoptosis will happen. So that's what happens in case of in the case of using UV lamp in the laminar airflow chamber. So in case of 70% ethanol, even when we are wiping it, we are calling it as disinfectant, right? So what happens is that this leads to lipid dissolution. That is, whatever the lipid membrane that are there, this phospholipid bilayer, that will get damaged. And as it's an alcohol, the cell will start like dehydrating. So once the cytoplasmic components are coming out, what happens? The cell shrinkage happens, which is nothing but your that will also lead to the cell death only. Okay, and protein denaturation also happens. So that is one of the reasons like why we are using 70% ethanol. So in that way also cell will get damaged, and using UV lamp. So it's just a combination that we are using it in the food. Moving to question 9, match the following parts of bioreactors with their purpose. Okay, sparger, as we have already seen that sparger is mainly used for It is being used in the airlift bioreactors for both the reasons. What? Aeration as well as agitation. So we can just see that, just a minute. It can be used for both the purpose, aeration as well as agitation. So as of now, we'll just see it. And here we have already been given this impeller. Impeller is nothing but agitator, right? If agitator is not being given, sparger can be given both the options. But the main purpose is aeration only. Agitation is the second purpose only in case of airlift bioreactors. So as impeller is already being given, agitation is the answer for that. And DO sensor, so which is nothing but to screen the oxygen level in the bioreactor. Okay. So the answer will be for sparger, it is aeration 2. For DO sensor, it is 4. And for your impeller, it is 1. 2, 4, 1 is the answer. So that is 241B. Okay. Moving on to question 10. Dr. X runs a bioreactor experiment and found that his process produces some toxic byproducts which are affecting cells' viability when accumulated. Which of the following mode of bioprocess is suitable to solve this problem? So whether it's A, batch mode alone, option B, continuous mode alone, option C, fed batch mode alone, option D, both fed batch as well as continuous modes. So if you see the, we know already that continuous can be one of the obvious reasons. So if you take any products like lactic acid, acetic acid, and all these, what happens when you do a batch experiment or something, when lactic acid being produced at a very higher concentration, the pH of the medium will drop. Because of that, the cell will only get inhibited. So, so this is called product inhibition. The product will inhibit the cell growth itself, and then the cells will not be able to produce further lactic acid. Okay, so in that case, continuous mode of operation is one of the preferred way, because when lactic acid or acetic acid is going to reach a specific concentration, that will be removed continuously from the medium, and the cells will be like recycled back into the medium. So we can maintain the product at a concentration that is not being toxic for the cells. Okay, so continuous mode is one of the obvious reasons, but yes, fed batch is also one of the main reasons, because if very high cell concentration is being for very high substrate concentration, for example 200 gram per liter of glucose is being given in the start of the medium, say for example. Okay, what happens is that this 200 gram per liter will be too much for the cells. So what happens is that the cells will start utilizing it for all the other unnecessary pathways also. Say for example, your pathway, like your product of interest is lactic acid, as the concentration is present very high, because of which the glycolysis will happen. And what happens when there are so much of cofactors, intermediates are already being present in the cells, like if the glucose is being utilized in the glycolysis, it has to go through some other pathway. But if there is any feedback inhibition, your product of interest, for example lactic acid, will stop utilizing further for the lactic acid pathway. So what happens is that this will get diverted. So already the acetyl CoA is being ready in your media because of the very high concentration of substrate. Okay, so it will be diverted towards acetic acid, formic acid, or else like any other byproducts will be diverted. Okay, so that's why fed batch is one of the preferred mode of operation. You can start feeding your substrate in such a concentration that it's not diverting it for all the other uh, toxic byproducts, or even not even toxic, even like byproduct formation also we can prevent by going for fed batch mode of operation. Okay, so if there is any question, you guys can post to me. Okay, thank you so much.